we can say what's the model we've got, and it turns out to be like this. And we notice, this is the point, it's of exactly the same form as we had before, but these coefficients now are functions of all these bits and pieces. And that's important because the disease model, we know what's going on. Here it's with a marker that's not the disease locus itself. So the true association model has the same form as the penetrance model, what's really going on. All right? And this is a remarkable thing. When this is not equal to 0, which is most of the time, the coefficient of the quadratic terms generally approaches 0 faster than does that of the linear term. What this means is, in practice, it means that unless your markers are very dense and highly correlated, it means fitting a linear model, which everybody's been doing, isn't a bad idea. You only want that quadratic term. It's only going to help you when your markers are very dense. That's what that's saying. And this is proved in an appendix. I'm going to give you the paper later on. Now, with all this machinery, we can do some power computations. Hotelling's t-squared in a retrospective model and the score test, the likelihood ratio criterion, likelihood ratio test in a prospective logistic model are expected to perform similarly. That's the whole point. So why don't we do it this way? The beauty is when we do it this way, when we can actually do something about power, we can compute the power analytically without having to do all those simulations. So that we need the non-centrality parameter for the t-squared for the two. If we take one with 5 things in it, now what we have here is a vector of 5 things, a vector of 5 things here, and here we've got a 5 by 5 matrix. There again, we're going to assume it has rank R, whatever it is, and this is how you get that. And so we take the generalized inverse, and then the non-centrality parameter for the other test can be obtained by taking subsets of that, whether it's a 3 by 3, 2 by 2, 4 by 4, what have you. And with all that, we've got the power is basically given by 1 minus with the R degrees of freedom now. We've got the cumulative density for a chi-squared with non-centrality parameter lambda evaluated at the value of the 1 minus alpha quantile of a chi-square distribution. The degrees of freedom in either case depending on what you've got in that big matrix. All right? First of all, let's compare the theoretical with the empirical. And what we did here was you've got these different models, additive, dominant, recessive, theoretical, t-squared, empirical, all these different tests, and you can see they're very similar to this theoretical one we got. We get this one by doing about 1,000 simulations and all certain parameters we had to put in. So there was we had 2,000 cases, 2,000 controls for recessive 500. Allow for some kind of Bonferroni correction. You have 500,000 tests, and we empirical comparison from the ratio, the number of rejected. We had 100,000 replicas. Okay, but you can see we can use the non-centrality parameter and get all these. Just now, we don't have to do any more simulation. First of all, let's look at the 1 marker tests. And we either have one with 1 degree of freedom, the allele frequency difference, or we put in both of them, or we use the Hardy-Weinman distribution, so that's 1 degree of freedom, 1, this is putting the 2 together. If your model is additive, then clearly this is going to be the best one, an additive model. If the true model is additive, your analysis model that says additive is going to be best. If it's dominant, it's not too much difference. Oh, by the way, notice this thing here is pretty useless. I thought it was pretty good at one point, but it's pretty useless. I thought it might be pretty good. But now if you've got a recessive model, that's when the 2 degree of freedom test starts kicking in, and even this one can start kicking in. And down here we've got how much disequilibrium there is. In other words, this is saying that covariance is large, correlation is large. This is saying it's 0. Right? As they get more and more correlated. Then you've got this one too, and I don't want to say too much about that. No one believes that one anyway, not even me. Well, it might be true sometimes, who knows. I personally would not spend a lot of effort looking at that, just to let you know. I think it's pretty unreasonable. Except if you're breeding corn or something, hybrid corn, hybrid figure, maybe for survival, but not as a disease model, I don't think. So now let's look at the 2 marker tests. This is where it gets interesting. So first of all, we showed that the type 1 error is about right. We've done all that, I'm not showing you here. So these are the various tests you can do. All right. We've got test 2.5, which says we have done 5 things, all 5 things in the model, we're going to test the 2 additive ones there. Only 4 things in the model, only 3 things in the model. This is a thing that geneticists like doing, a haplotype-based test. Very complicated, because they can't find these things. Well, they do, they have a software that tries to get at them. And this is test 2.2, and this is the LD contrast test. Now this basically says we have a mild amount of correlation between successive markers and between our marker and our disease. This one says we have stronger disequilibrium, stronger correlation. Right? What I want you to notice is, first of all, that the best, bold is the best test, all right? Here, 2.4 and 2.2, these are 2 additive things. They look pretty good. When it gets very dense, you also find that at this point 2.3 comes along. So that's all very well. Let us look at some semi-real data. Semi-real. And what do I mean by that? Well, it's going to be simulated data in the sense that we are simulating who has the disease and who doesn't have the disease according to a particular marker. But all the other markers are there. They're true. They're found from what is known as the HapMap. Okay? We've got the HapMap CAU population, estimated the LD parameters, that's the correlations. Data consists of 120 haplotypes, estimating 30 parent offspring trios. Split chromosome 11 into mutually exclusive consecutive regions, each containing 3 of these things. And we estimated the correlations and allele frequency parameters. We excluded regions where the minor allele frequency of the 3 consecutive was less than 0.1. People do that because they say it's not very accurate, so if all 3 of them throw those out. And that left us with 4,648 millions. Right? We chose the one that had the smallest allele frequency amongst the 3 to be our disease, and we simulated the disease on it. So these are real genetic data, but you simulate the disease according to one of these models. So parameters, the same as before, we had a whole bunch of parameters. Okay, now what do you get? So this is what you see now is you'll notice that the 2 marker test that is best is always better than the single marker test that is best. See, that is the power. All right. Furthermore, you will see that sometimes this is the best, sometimes this one, sometimes this one. Overall, I suppose you could say this isn't too bad, that's not too much smaller. That's not too much smaller, and that's not too much smaller. What we're going to and this thing never works. I had great hopes for that. I thought this is what we should all do, but this shows it doesn't work. I thought that's what we should do. All right. So we're going to see later on that this test, at least for the Wellcome Trust data, works remarkably well. So just recently, we've tried looking at the Wellcome Trust data. Real data. So the Wellcome Trust Case Control Consortium, they had data on 2,000 cases with coronary artery disease, 2,000 cases with hypertension, and 3,000 shared controls. They actually studied 7 diseases. We look at these 2 because they found absolutely nothing for hypertension and just one thing for coronary artery disease. And we said, well, let's say to a single market, do 2 at a time. Notice when we do 2 at a time, we don't double the number of tests. In fact, we do slightly less because of the end effect. My reviewers as well, we're doing lots of, no, same, all right, no problem. QC, quality control, same as they did, all right. Repeated the 1 marker test just to show that we get a similar answer because we're using the logistic regression, they use the, whatever they called it, the trend test, Armitage-Cochrane trend test, Cochrane-Armitage, however you want to put it. And we actually also, because when we submitted this paper, they said, you don't know, you've probably got heterogeneity, so we put in a lot of principal components. Those who know what I'm talking about, you know what I mean. They got the same answers, basically. So then we did these, okay? We did the 2, 3, and the 2, 2, including the first 10 principal components of the base. Because I did these principal components, they got from all the genomic data all together, and it gets rid of what we hope. It gets rid of the fact that there's heterogeneity. That's what it's meant to do. I can't go into it in detail, but that's the idea. So this is what happens. Results. We found that the 2, 2, and the 2, 3 test for the Wellcome Trust data, that's 500,000 markers, roughly evenly spread across the genome, gave essentially the same result as the cross-product term usually being non-significant. And in 1 case did it actually bring in more. But typically the 2, 2 was fine. In other words, those data, being only 500,000, are not dense enough to make use of the cross-product term. All right? Maybe if we had 1 million, or now we're getting more than 1 million, maybe then it would be useful. But those data didn't seem to help that much. All right? The correlation in some of the pairs was close to 1. Now you'd have thought if their correlation was close to 1, surely doing a 1 at a time would find it. But it didn't. Worried us a little bit until we figured out what was going on. Or they did, the thing is, even though they, and people, typically when people do this, they say, well, if the two things are correlated greater than 0.8, we throw 1 out. Bad idea. Very bad idea. I think you should wait till 0.95 or 0.98 before you throw 1 out. Get much more information. Right? Yes? Oh, keep by the microphone. Thank you. All right. So what we did is, with a lot of work, computational work, we obtained the phased haplotype tables to check by Fisher's exact test that the small p-values were not due to just small sample sizes. And I feel fairly confident that is so. Now, one of the reviewers brought up the fact we really ought to look at the clusters, the plots, and all that stuff, which is unrealistic if you try. I mean, this would take another year for us to get the data from the Wellcome Trust, blah, blah, blah. So we wanted to skip that step.